Yes, so this is the latest, one of the latest biology past exam papers, October, November 2022. All right, hold on. Paper 4, we can take a look. First question, related to topic number 1. So here, phytoplankton consists of many species of single-cell and many-cell algae. Algae are classified in the Protist kingdom. All algae contain one or more chloroplasts. State the name of another kingdom that contains organisms which have chloroplasts. So you may want to have a quick revision regarding the kingdoms. Hold on, let me search the information. Yes. So we have the Protist kingdom here. Yes. And then we have monarans, fungi, plants, and animals. These are the 5 kingdoms that include all the living things on Earth. So this is the most general categorization. For IGCSE, we mostly focus on plants and animals kingdom. So we will focus mainly on these 2. Yes. So we can see here, algae is Protist kingdom. All algae contain one or more chloroplasts. Name of another kingdom that contains organisms which have chloroplasts. This will be plants. Plants have chloroplasts for photosynthesis. Next, student investigated the species composition of the phytoplankton in a lake. Here, we show some of the phytoplankton collected by the student, phytoplankton samples. So now we focus on X here. The actual length is 0.19 mm. Convert the actual length of alga X to micrometers. So in this case, we need to do a unit conversion. This is quite straightforward. Then now we need to know that 1 mm equals to 1000 micrometer. So we can finish the calculations. 0.19 mm multiply this conversion factor here. 1 mm equals to 1000 micrometer. Complete the calculations that will give us 0.19 mm equivalent to 190 micrometer. All right. Next question. Now, student made careful drawings of 7 types of algae found in the samples of lake water. So we have 7 types of algae with different shapes, different appearance, and so on. Next, what we need to do is we need to use the key to identify each of the species A to G. Write the letter of each species in the correct box beside the key. Now I already copied the pictures to this page here, so we can take a look straight away. Alga has 4 spine-like extensions. 4 spine-like extensions. If we look at our options here, we can see straight away F. 4 spine-like extensions. We can see this. Next, the other one that have these characteristics, this here. 4 spine-like extensions. So we have these 2 candidates here that fits the description for this 4 spine-like extensions. So we go to number 2. Alga has spine-like extensions that are all the same length. We can straight away relate to this here. The spines have the same length. So we expect the answer, it will be G. Straightforward. First one, G. Next, alga has spine-like extensions that have different length. So, straightforward, this will be F, different lengths. So, next answer will be different lengths, F. Next, coming back to here, alga that does not have 4 spine-like extensions. Going to 3 here. Alga has a round shape. Straight away, we can relate to this here. Round shape. Straightforward. B. Next. Alga does not have a round shape. Go to 4. So now we go to 4. Alga narrows towards the end. So the end points are narrow. So we have 2 candidates here. C, that have narrow ends, as well as donkey that has narrow ends. Going to 4. Oh, sorry, going to 5. 5, alga cell is semicircular. Semicircular, straight away, we can relate to donkey. So the answer here, it is donkey. Next, algal cell, not semicircular. Straightforward, it will be C here. Narrow towards the end, but not circular. So the next answer will be C. Moving down the pictures. Okay. Almost done. Alga is branched. This is straightforward. Straight away, we can relate to A. Branch. So next answer, A. Finally, alga is not branched. Straight away, we can relate to E here. Straightforward. Final answer, E. All right. Next question. Phytoplankton are photosynthetic organisms. Means they carry out photosynthesis. Describe the importance of phytoplankton in the food web of lake ecosystems. Based on its characteristics of being able to carry out photosynthesis, so it can play a very important role in the food web of the lake ecosystems. We can take a look some of the main points here. First, they can act as producers. Through photosynthesis, they can produce food. So they act as producers that provide food to the other living things in the food web. Essentially, so this is a 3-point question. So any points, any 3 from these points here will give us the full marks. Yeah, so basically we talk about the, we explain each accordingly. So because it can carry out photosynthesis, it converts sunlight energy into chemical energy. Essentially, provide a source of food for primary consumers, such as herbivores. We can also call that second trophic level. So if we were to give the name of the food, it's going to be something like glucose or starch, something like that. So these food sources will provide energy to other trophic levels or the rest of the food chains. So the other animals, such as the omnivores or the carnivores, they can consume the herbivores to get the energy. Yes. Yeah, so these are all of the main points, or one of the other minor points, we can say that basically the process of photosynthesis will basically convert water and carbon dioxide to glucose, which is the food sources for the remaining of the food web. Next question. So this is a question about the digestive system. So we have the human alimentary canal and associated organs. So the organs that are responsible for different functions in the digestive system. So here we have a table that shows enzymes, the organs that secrete these enzymes, their substrate and products. Yeah, so basically some of the main components we need to pay attention in this chapter, digestive system. So we can go through one by one. So I have already copied the picture here. So first, enzyme amylase. We know that amylase is produced at two places. First is the mouth. Secondly, the small intestine. So here, our identifying number is 1. So we are talking about the mouth mainly. So organ that secrete the enzyme will be the mouth, the salivary glands in the mouth. So the substrate of amylase in the mouth will be mainly starch. So it will break down starch to produce maltose. So essentially, the long chain of the starch will be broken down into units of 2. Units of 2, units of 2, units of 2 of the glucose. And then final, so each unit, we call them maltose. They are made out of 2 glucose. So in the final step of the digestion, the maltose will be broken down to individual glucose molecules. Next. So here, next, the information that we have is number 3. Number 3 is related. We can see stomach here. So we are referring to the stomach. The substrate is protein. That makes sense. We know that the main function of the stomach is for protein digestion. So we're going to take a look one by one. What is the enzyme responsible for protein digestion? We know that it is pepsin or protease. Organ that secretes the enzyme, straightforward, the stomach or the gastric gland. Finally, the products. The protein broken down into amino acids, straightforward. Next, we are looking at number 4. Number 4 is pointing at the pancreas. We can see that it is pointing to the pancreas. So lipase is produced, the organ will be pancreas. And then the substrate. What are we breaking down with the lipase? We are breaking down the fats or lipids or oils. And the products will be, once we broken down, we will end up with fatty acids and glycerol. Next, here we focus on, okay, so what are we focusing on? This one. We have maltase, organ that secretes the enzyme, small intestine. So the number will be 5 here, straightforward. And then substrate. Just now we already mentioned here, breaking down the maltose. The function of maltase is to break down maltose into individual glucose molecules. So our answers for this part will be maltose and glucose. Next, explain the role of hydrochloric acid in the alimentary canal. 3-point question. So we know, we are very familiar. Hydrochloric acid produced in the stomach. So the main functions will be, any three will be acceptable. So yes, the reference that hydrochloric acid produced in the stomach will be 1 point. Secondly, it kills bacteria and pathogens. So one of the main functions, highly acidic, very corrosive, very damaging to the bacteria and pathogens. It can denature enzymes in bacteria and microorganisms. So one of the ways it kills the bacteria or pathogens by denaturing the enzymes in the bacteria or microorganism. Provide acid or suitable pH for the enzyme reaction. So we know that in the stomach we have protein digestion. Protein digests to form amino acids. So the enzyme responsible for that will be either hepsin or protease. So for hepsin and protease to the optimum pH will be acidic. So the hydrochloric acid will provide this optimum pH for the digestion. Next, a function of the region of alimentary canal called label number 6. Coming back to here, number 6, we can see here, we are pointing at the large intestine. We are very familiar with that. So in the small intestine, we completed all of the digestion process and then we absorb everything that we need from the food. So the remaining substances that are non-digestible, that we cannot absorb, or waste materials that we don't want, will be passed into the large intestine. So what happens there? You can see here, reabsorb water, ions, or vitamins. Main activities, main processes that happen in the large intestine. For another possible answer, fermentation of indigestible food matter by bacteria. Next, this is a long response question. We can see that it is a 6-point question. So what we are required to do here, describe how food is eaten by humans. How food eaten by humans is reduced to smaller pieces, and explain how this makes chemical digestion more efficient. So essentially here we are talking about physical digestion or mechanical digestion. So here our main objective is just to turn the food particles into smaller pieces to increase the surface area for the upcoming chemical digestion. So in this case, we are not having any chemical changes to the food particles. We are not breaking or forming any new bonds here. So no chemical changes at this stage. So later, when we have the chemical digestion, then we start to have the chemical changes whereby we involve breaking bonds, forming new bonds, form new compounds. So that's how we differentiate physical and chemical digestion. So the points that are acceptable as answers are a lot. So any 6 that included will get us the points. Okay, so first, the point that we are talking about mechanical or physical digestion. So the main function is to decrease the particle size. When particle size decrease, surface area of food particles will increase for any activity. So at this point, any enzyme activity. So, for example, if we mention any enzymes, for example, let's say we mention amylase to improve the amylase enzyme activity. So essentially, decreasing the particle size will increase surface area, and then increasing surface area will be beneficial for any enzyme activity. So for example, here we can mention amylase, pepsin, any enzyme, then we can get one more point here. Any further detail of enzyme activity also will be acceptable as a point. So at this point, we are talking about how it happened. Mechanical and physical digestion, how it happened by chewing, crushing, grinding, by using our teeth. Okay. So another good thing is by mentioning different types of teeth and their function will get us points as well. Incisor and canine used for cutting food based on their shape. Molar and premolar used for crushing the food. And then another type of mechanical digestion will be mixing of saliva with food. So yeah, so basically this will mix the food evenly with, for example, with our saliva, and inside the saliva there will be amylase enzyme. So that will make it easier for the amylase enzyme to act on the substrate. And then formation of bolus that make it easier for swallowing. Another mechanical digestion that happen is in the stomach. So what happens in the stomach? So we have a lot of muscles in the stomach that can churn, okay, that can keep churning and then keep moving around the food. So basically mix well with the liquid in our stomach, and also mix it well to maximize the enzyme action. So basically we will form the food that we eat will become a liquid mixture called the chyme. Yeah. So basically how it happened is due to the muscles in the stomach. Okay. And then another type of mechanical digestion that happen is emulsification of lipids. So the fats and lipids that we consume, they are non-water soluble. But in our alimentary canal, it is mostly water. So we need a good way to increase the surface area of the lipids so that the digestion will be more effective. So we will have the emulsification by bile. So basically, bile is made of salts that is produced by our body. Yes. So this process will occur in the small intestine mainly. So the presence of the bile, it will break the fat globules into smaller pieces. So large fat droplets will be turned into smaller droplets. So this again, this will increase the surface area. So all of this process, okay, these are all the physical digestion processes that happen in our alimentary canal that will help the chemical digestion to become more efficient. Next question here. So in the picture, we can see aphids. Picture of the aphids. They are insects that feed on phloem sac by inserting their mouthparts into phloem tissue. We know that the function of the phloem tissue is for sugar transport. So yeah, so the sugar is produced by photosynthesis, and then the sugar is transported through the phloem to different parts of the plants. So yeah, so it makes sense. So the aphids, they yeah, so basically they get access into the phloem okay, by inserting their mouth parts into the phlegm so that they can get to the sugar. So here we have a picture show aphid feeding on phlegm tissue in a leaf. State 2 features visible in 3.1 that identify this animal as an insect. So we recall the characteristics of insects. So these are the main points here. Characteristics of insects. 3 body parts. Head, thorax, and abdomen. So 3 body parts. Head, thorax, and abdomen. So main characteristics of insects. Secondly, 3 pairs of jointed legs or 6 legs. Straightforward, we can see that. Yeah, so these are the characteristics of insects. Next, aphids can transmit viral pathogens when they feed on a plant. Okay, so they can carry virus. Okay, so basically, yeah, so the virus can infect the plants and damage the plants. How a severe infestation of aphids on crop plants can be avoided. So basically, they can be damaging to the plants. So we need to think of some things that we can do to prevent the aphids from the virus on the aphids from damaging the plants. We can take a look. 2-point question. So any two will be acceptable. So main method, fast and effective, will be spraying insecticide or chemical to kill the aphids. Secondly, we can grow the crops in a glass house or protected environment. Yeah, so growing in the glass house so the aphids cannot get in there as a protection. Use crops that are resistant to insects or aphids. So we can have selective breeding or genetic engineering to produce the breeds of the crops that are resistant to the insects. Yes, yeah, we can use the method of selective breeding or genetic engineering. Okay, other methods, biological control. So for example, maybe we know some birds, okay, or other insects that feed on the aphids. So we can have these other predators in the environment to kill off the aphids. Okay. Another possible point, avoid monoculture. So if we have monoculture, means we have all of the same crops on one plot of lands. So this is not that ideal because we will use up all of the nutrients in that plot of land. So we need to keep changing to different crops so that we can replenish the nutrients that's in the plot. So plant a variety of crops. For example, do a crop rotation. Next, aphid shown in figure 3.1, feeding on phlegm tissue in a leaf. So that's what we are seeing here. State the names of 2 tissues in the leaf that the mouth parts of the aphid pass through to reach the phlegm. So for this question, it will be useful to take a look at a picture. Hold on. So this is a cross section of a leaf. So the aphid is here somewhere. So this is the aphid here. So it's trying to reach to this section here, the phlegm. So it's trying to reach to here. So all of the tissues of the leaf that the mouth parts pass through will be any of these. Any of these will be acceptable answer. So the mouth parts need to pass through all of this tissue to reach to the phlegm. So that's the idea. We can take a look. So yeah, so any of these will be acceptable answer. Upper epidermis. So this one here. No, this one is cuticle. Upper epidermis. Xylem. So it needs to pass through the xylem. So we'll reach the xylem first and then phlegm. And then palisade mesophyll, also one of the layers that need to pass through. Spongy mesophyll, also one of the layers that need to pass through. So that's the main idea for this question. Next, so we have another experiment that is going on here. So what is going on? We have aphids have been used to investigate the translocation of sucrose in phloem tissue. While they are feeding on phloem sap, aphids excrete a sucrose-rich fluid known as honeydew. So essentially, we have two groups of aphids here. So they will feed on the sugar in the phloem, and then they will produce another liquid called the honeydew, and then the honeydew will be collected on the petri dish. So that's the idea. So by checking the contents in the petri dish, we can find out essentially how fast the sugar is traveling. Yeah, so we know sugar is transported by the phloem. So we want to find out how effective the phloem is as a transport vessel. Yeah. So how fast it takes to get from one part of the stem to another part. Yeah, so in the investigation, we have two groups of 4 aphids were placed at intervals along the stem. Yes. So here, the leaves were enclosed in airtight glass chamber. Okay. A special form of carbon dioxide gas that contain radioactive carbon-14. So we have the CO2 made of carbon-14 here, supplied to the leaves for a short period of time. So this is quite useful because the carbon-14 is radioactive, so it can decay. So it can decay, so means that we can detect the carbon. We can detect the carbon-14 here, and then we can detect the carbon-14, how much at each location. So divided by the time, we can work out how fast the sugar is traveling. Samples of honeydew collected at intervals. So we collect the honeydew here. Then the time taken for sucrose containing carbon-14 travel the distance between group A and group B was recorded. And then we repeat the investigation. Investigation repeated twice using a fresh stem at different groups of aphids for each trial. So we do repeat the experiment. So now we can see the results here. So we have 3 trials here, 1, 2, and 3. So the main thing, so this will be our independent variable here. Independent variable. So what are we investigating? Distance between group A and group B on the stem. So distance between group A and group B. So this is quite reasonable. When we increase the distance, it will take a longer time for the sugar to travel straightforward. That's the main thing we are investigating. So different distance between group A and group B. So this is the stem here. So we have group A of the aphids and group B of the aphids over different distances, 650, 340, 630, and so on. And then we can see here the time taken for the carbon-14. So in here, essentially, we are talking about the sugar that contain the carbon-14. So the longer the distance, the time taken will also be longer. So yeah, we get the answer, the result is what we expected. So the dependent variable. The dependent variable here is exactly what we expected. So longer distance versus shorter distance. the time is also shorter. The rate of movement of the carbon-14 mm per hour. Rate of movement. So how much carbon-14? So means we are talking about how much sugar travel across the stem per hour. So how fast is moving? Distance per time, mm per hour. So yeah. So for the trial number 3, first 2 is calculated, provided for us. So for number 3, we need to calculate by ourselves. So based on the information given, we calculate accordingly. So rate of movement will be essentially distance, how fast. We are talking about how fast it is moving. So distance divided by time. So here we have distance mm, time minutes. So we have mm here and then minutes here. But we need to be careful about this. So here we have mm per hour. So means we need to change the minutes to hour. Yeah. So, okay, so we can take a look at the calculations. Okay, so we have here 630 mm and then 150 minutes. So 630 mm, it is in the correct unit, no problem. So 150 minutes, we do the unit conversion. 1 hour equals to 60 minutes. Yes, so completing all of the calculations, we can see that the final answer will be 252 mm per hour. Yeah, so that's our answer here. Okay. Yeah. Okay. Next. Outline how carbon-14 in carbon dioxide gas becomes incorporated into the sucrose molecules that are translocated in the flume. Okay. So how it started? We start as a CO2, but the CO2 is made of carbon-14. So we know CO2 through photosynthesis. Through photosynthesis, it will be converted to glucose. And then glucose will be converted into sucrose. Yes. So glucose is a single unit of sugar. Sucrose is what we call disaccharide. So 2 units of sugar is more suitable for transport purpose. So the sucrose is the one that will travel in the flume. So that's the main idea. 3-point question. So we will look at the answers here. Okay, so any 3 will be acceptable. So what happened? We know first carbon dioxide enters the leaf through stomata. So first point. So we talk about how it enters the leaf from outside air into the leaf by diffusion. We have the picture just now. So it enters the leaf through the stomata. Through the stomata here, entered the leaf by diffusion. And then, so basically it can travel, okay, to the palisade mesophyll, the main site where photosynthesis happen, also in the spongy mesophyll. So photosynthesis can happen, and then glucose is produced. Another point. So these are the main points will be 1, 2, 3. So this will be the main points. So another related point, photosynthesis catalyzed by enzymes. And then photosynthesis occur in chloroplast. And then next, glucose converted into sucrose, which is more suitable for transport purpose. Next part. Sucrose travels from stem to other parts of the plants known as sink, where it is used. 2 parts of the plants that act as sink. So for the plants, the glucose produced here in the the region with the leaves. And then sucrose produced will be transported to other parts of the plants for storage purpose or to be used and so on. So what are the other parts? So all of these will be acceptable answers. Buds, roots, tubes. Buds and roots, they are actively growing. So we provide the energy for it. So that will be one of the things. Okay. Tubers. So tubers at the root region, we have the tubers, okay, that can store up the excess glucose, okay, or flowers, okay. So flowers, fruits, seeds. So these parts can also accept the glucose for storage purpose or for growth purpose. Young or growing leaves, yes. Shoots, okay. And also to produce nectaries, to produce honey. Next, sucrose is used in the cells of the zincs in the plants. Describe uses of sucrose by zincs. Okay, so just now we talk about the different parts of the plants that act as the zinc, and then now we talk about how they are used. Okay, next. First, sucrose can be converted to glucose. So sucrose itself is is not that active. It cannot be used directly as an energy source. So it's more suitable for transport. So when we actually want to use it, then we will convert to glucose. Then it can go through the respiration process to produce the energy. Yeah. So essentially, we can use the sucrose for any plant process that requires energy. So any plant process that requires energy will find uses in this sucrose. For example, can be used for growth, reproduction, flowering, active transport, absorption of ions, cell division, mitosis, metabolism, formation, and so on. Yeah, so this is great. So essentially, all of the important processes in the plant will need energy, and the sucrose can provide for that. Another thing that it can be used to be converted into starch for storage purpose, or to produce cellulose for cell walls, or converted into amino acids for to produce proteins. Yes, or another just now we have mentioned to make nectar or honey. Next question. So here we have a flowchart showing some of the processes that occur in a biofuels power plants. Okay, so we know that biofuel is very important, okay, because it's so basically we take some of the plants. So some of the tissues of the plants, the stem, the leaves, okay, something like that. Something that we cannot use as the food, okay, then we can convert it into biofuel to produce energy. So this is another, we can say, environmental friendly way to produce the energy. So when we cannot eat it as food, we can convert to energy to be used. Okay, so example, we can take crop waste, because let's say we grow the rice plants, but only the rice can be eaten, but the rest of the plant, the leaf, the stem, all we cannot eat it. So this becomes the crop waste, but we can use it. Forestry waste, if we, for example, cut down the tree, only some parts of the trees can be used, then the rest will become waste. We can use that. A giant reed plant grown for biomass. So we can also find some type of plants that are very suitable to be converted into biofuel. Okay, so we will treat it. Okay, it will undergo some treatments, and then it will be release complex carbohydrates. So the carbohydrates we know contain the carbon. So the carbon can be used as energy later, break down by enzymes, then it will produce sugars and glucose. Fermentation by yeast, it will produce ethanol. So the ethanol will be the biofuel. Okay. So the fuel that is coming from biological sources, biofuel. Fermentation stage requires yeast. Complete the balanced chemical equation to show how ethanol is produced by yeast respiration. So you may recall this is about anaerobic respiration of yeast will produce ethanol. So the main components of the equation. So we start with glucose. So we want chemical equation. So chemical equation means we need to recall the chemical formula. So we cannot give the word equation. We must give the chemical formula. So sugar will produce ethanol. So 2 molecules of ethanol and then carbon dioxide. Okay, so this is how it happens for the anaerobic reaction. Using the information in figure 4.1, suggest the environmental advantages of using ethanol as a fuel. Okay, so yeah. Okay, so referring to this picture here. So these are the main points that we can mention. So any three will be acceptable. So environmental advantages, okay? So why it is good for the environment? So first, it uses the waste from farming or forestry. So this is quite good because we can use the waste and turn it into something useful, okay? Second, it will reduce the use of fossil fuels, okay? Or non-renewable sources. This is quite good because we know fossil fuels, they are not that environmental friendly. Okay. Yeah. So if we use more biofuel, we can reduce the use of fossil fuels. So that is good. Less pollution, damage to environment by mining or drilling because we need to mining or drilling to get the fossil fuels out of the grounds. So this can cause pollution or damage to the environment. So if we use more biofuel, this will be reduced. Conserve fossil fuels non-renewables for the future because the things like fossil fuels, the amount is limited. They are non-renewable. So if we use less now, we can save some for future use. That's good. Plants grown for biomass is an example of a sustainable resource. So instead of taking from the fossil fuels, we take from the plants. Okay. So the plants, we can plant the new plants. So if we need more, we can plant the new plants. So that's what we mean sustainable. So we can replenish. We can get continuous supply if we need to. Carbon neutral or small carbon footprint. Okay. The idea is, okay, this, if we burn the fossil fuels, okay, it will produce carbon dioxide. So it's actually damaging to the environment because it causes global warming. Okay. So CO2 can cause global warming. Okay. So this is not good. But if we use biofuel, it is carbon neutral. Why? Biofuel, okay, if we burn the biofuel, we burn the ethanol, okay, we will get CO2. Yes, we also get CO2. Same problem with fossil fuel, but no problem. Why? Because now we will be planting the new trees because the biofuel is coming from the plants. So after we burn this CO2, we will plant the new plants to take in the CO2 again. So this is how we determine that it is carbon neutral because fossil fuels, we take it from the ground, we mining it, we take it from the ground, we burn it, we form the carbon dioxide. It's quite bad. Okay. But if we get it from the biofuel, it's actually carbon neutral, because at the same time we are planting new plants that will absorb back the carbon dioxide that is produced here. Yeah, so it's good in that way. Next, so because it is carbon neutral, it does not contribute to the greenhouse effect or global warming. So at the same time we produce CO2, at the same time we are planting the new plants to take in the CO2. Less air pollution or no acid rain, because if we use more biofuel, we will use less fossil fuels. When we use less fossil fuels, okay, we know that in the fossil fuels we have nitrogen and sulfur. Sulfur and nitrogen will produce nitrogen oxide, okay, or sulfur oxide. So this will cause acid rain. So if we reduce fossil fuels, then we can reduce the acid rain. Yes, okay, so these are all of the acceptable points. Next, farmers grow giant reed plants as monocultures. So just now, we had a look here. So the sources of materials that we can use for the biofuel, we have three sources: crop waste, forestry waste. So we already know that, so we can use the waste material for the sources for the biofuel. Another thing that we can use is we are not using the waste material. We specifically plant some type of plants that can be used to produce the biofuel. So giant reed plant in this case, okay? Monoculture, meaning that we select a plot of land, okay? We think that this reed plants is good to produce biofuel, okay? So maybe it's like very efficient to be used to produce biofuel. But we plant as monoculture means the whole plot, we plant the same thing, okay? Monoculture. So this can be a potential problem, okay? Describe the disadvantages of growing giant reed plants to provide biomass for the production of biofuels, okay? So just now we discussed about using the waste material for the biofuel, okay? We think that is a good idea, no problem. How about we choose a plant specifically to, it's not a waste material, we just purposely plant these reed plants to produce a biofuel. Can that be a good thing? Okay, what are the potential disadvantages? Okay, we can take a look. Okay, so some of the problem is we use up this land to plant these reed plants, but the land could have been used for food production. So this could be a bad thing because we occupy this land for the reed plants. So this plot of land could have been used to plant the food. So that could be a bad thing. Loss of habitat. So we clear out the land. Let's say we clear out the forest for these reed plants. So the forest is gone. So the animals the animals and plants from the forest, they are losing the habitats. Okay. So they may die off. Okay. So all these living things that's supposed to live on that land, they will die off. So we will lose diversity. Okay, so some of the species may be extinct and so on. Loss of genetic diversity related. Okay, yes, and then the land could have been used as nature reserve or conservation to protect the different living things there instead of clearing out and then use for this. Okay, so monocultures. Problem with monoculture, susceptible to pests and disease. Okay, monoculture means they all the same type of plants. So they are, so let's say we have a virus or we have some insects that attack the plants, because they are all the same plants, they will die off. Okay, all of them, because they are not resistant. Okay, loss of soil nutrients, soil degradation, soil erosion. Yeah, so if we keep using the same plot of land for the planting without taking care of it, the nutrients will be lost. Okay, and then we need to put in a lot of chemicals into the land to maintain it. For example, we need high input of herbicide, pesticide, water, fertilizer to grow this red plants. So that could be a high cost, or it could be a problem. It can cause pollution and other problems. Yeah. So this is one way, okay? It's quite useful. We think about a problem from two perspectives, not just the advantages. We also think about the potential disadvantages. Okay. Next, one problem with using biomass in the process shown in figure 4.1 is the breakdown stage produces a sugar called xylose and ethanoic acid. Yeast cannot use xylose, and ethanoic acid is toxic to yeast. Scientists genetically engineered a type of yeast that can use xylose and ethanoic acid. So we have this figure here, shows result of one of the trial experiments done by the scientists using the new genetically engineered yeast. Okay, so first we have so to do the anaerobic respiration, we are converting glucose to ethanol. So that's what we want. Okay, so we use the yeast. Okay, that's good. But this biomass that we have, the waste, the crop waste, and also the reed plants. Okay, so after the breakdown, they can produce glucose as well as xylose. Okay, but the yeast, they can use glucose only. So that's not very effective because we also have the xylose. So what we want, what they're trying to do is they go through genetic engineering, okay, to produce a new type of yeast. Okay, the new type of yeast is quite good. It can use xylose and also ethanoic acid. Okay, so it can also use xylose and ethanoic acid to produce ethanol. Okay, so that's quite good. Okay, so we can take a look at this picture here. What are we looking at? Okay, so if we are using this new type of yeast, okay, here. So what we expect to happen? First, we can see, okay, cool. That's good. So first, we know that it's supposed it can use xylose and ethanoic acid as the substrate to convert to ethanol. Okay, so this is xylose here is decreasing. That's good. That's what we want. Okay, and then the ethanoic acid is also decreasing. That's what we want. That's good. And then this is the ethanol. Okay, so ethanol is produced. Okay, that's good. No problem. Okay, that's what we expected, what we want. Okay, the substrate decreases and the product increases. Okay, describe the results shown in figure 4.2. Okay, so this is a 3-point question. So basically we need to the main thing is to describe the major trends happening here. Okay, so these are all the points, acceptable points. Okay, so first one, concentration of xylose and ethanoic acid decrease. Okay, this is good. This is what we expected. Concentration of ethanol increase. This is also what we expected. Okay, the reactant decrease and the product increase. Okay. Steep decrease and of xylose and ethanoic acid until 62 hours. So first, this is a general trend. Next, to get more points, we need to go into details. So we can mention like any of the details that we can identify with numbers. So we quantify the changes, then it is clearer. So yeah, so we can see a steep decrease up to this point here. So we can see like very steep decrease here. So means the rate of reaction is quite fast at this point. Yeah, so the steepest change is between 6 to 12 hours. Okay, so we can see here the steepest change between 6 to 12 hours. Okay, some of the details we can mention. Increase in ethanol concentration to 44 gram per dm cube. So we can see here, finally, the amount of ethanol produced is 44 grams per dm cube. Okay. Small change in concentration after 62 hours. So at first, it's quite fast. The reaction is quite fast. And then it slows down. And then it, so basically it's almost stopped at this point here. So small change in concentration. So xylose reaches 1 gram per dm cube. So xylose increase, it decreased until it's close to 0. So it means we almost use up all of the xylose. It's good. We use up all of the reactants at 90 hours. Ethanoic acid reaches 0.2 gram per dm cube. Ethanoic acid reaches 0.2 gram per dm cube. So that's also quite good. Okay, we almost use up all of the starting materials. Yeah. So basically what we need to be careful is to read the X's carefully. For example, this one is for the xylose and the ethanoic acid. And then this one is for the ethanol. And then this axis here is for the ethanoic acid. So we need to be very careful, like make sure we are reading from the correct axis to get the answers. Okay. Next. Okay, experiment done at 30 degrees Celsius. Scientists repeated the experiment at 20 degrees Celsius. Predict the result you would expect for the concentration of ethanol. So we are very familiar with this. When we decrease the temperature, so basically the rate of reaction will decrease, okay, because of the temperature decrease. So when rate of reaction decrease, we expect less ethanol will be produced. Okay. Yes. Okay. So slower rate of increase for ethanol or less ethanol. Take longer to reach the same concentration of ethanol. Okay. So the line of the gradient will be less steep. Okay. Yeah. Next. Okay. So here we can see the figure showing McKinley's cuckoo dove. Okay. We have the scientific name here, which is found on most of the islands in the southwest of the Pacific Ocean. Okay. So we can see this bird here. Okay. So Kaka Island is one island where McKinley's cuckoo dove is found. This species is part of many communities that are adapted to the different habitats on the island. Okay. Yeah. Okay. So basically, this Kaka Island, it has like specific climate, the temperature, okay, the rain, okay, very specific environment, okay, or it have like a certain kind of trees, okay, certain kind of environment, okay, whereby this bird, okay, this dove is adapted to live here, okay. So some features of the bird that make it suitable to survive in this environment. Okay, so define the term community. Okay, yeah, so we have a number of terms that we need to know about this topic. Yeah, so one of them is community. Okay, so what does community mean? Okay, all the populations of different species in the ecosystem. Population of different species. So this will include animals and plants. Okay. Complete the passage with the most appropriate word phrase. Okay. Adaptation. What it means by adaptation. Okay. The process resulting from natural selection. Okay. By which population become more suited to their environment over many generations. Okay. Because the environment itself, it does not change. Okay. So basically it remain like pretty constant. So for the animals or the plants to survive in this environment, it needs to change itself. So that's the idea of adaptation. So it need to change itself so that it's suitable. Its features will become suitable to survive in this environment. So yeah, so basically it happens quite slowly over many generations through natural selection. Okay, natural selection means that like for example, you have like different species of bird. Okay, different species of birds. So some of them will be more suitable to survive. Then they will survive. Okay, some they cannot adapt. So maybe like the weather is too hot or too cold for them, or there's no food for them on this island. So they will die off. So this is the idea of natural selection. The species that have the suitable feature to survive in that environment, they will survive. Okay? They will be selected by the nature to survive. Then they will continue to breed to the next generation and so on. Okay? But the one that die off, they no longer continue. So over many generations, we only left this, the one that can adapt to remain in this environment. Okay, so here we have another set of data. Highest point on Kaka Island is 1,800 meter above sea level. Okay, yeah. So, okay, so this is our plots here. Okay, so the island, okay, so we are given, okay, so this is the island. The highest point in the island is 1,800 meters, okay, above sea level, okay. 1969. In 1969, we have a researcher survey the bird species on Kaka Island. Okay, so basically we have this researcher. Okay, so basically it just looking at the looking at the different species, observing the different species. Okay, in the on the island, different species of the birds. Okay, he recorded the vertical distribution of the birds between sea level and 1,600. Okay, yeah. So basically, he's trying to see like where 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 do the birds um fly? Okay, where the birds spend their time. Okay, so not all the not all the environments are suitable for the birds. Yeah, so maybe like for some of the birds, okay. Yeah, so some of the birds, they may prefer to stay at a higher position of the island. Okay, so some of the birds prefer to stay at a lower position, but some birds, they can be anywhere. Okay, so for example, these three groups, they more prefer to be at this region here. Okay, so maybe they can find food, they can find safety. Okay, for example, so they mostly spend their time here, okay, at the higher position. And then we have this cuckoo dove that basically can be anywhere. Okay, any height is suitable for them. Okay, so these are the information that we can see so far. Okay, yeah. So in 2013, researcher repeated the survey. So this thicker line is 1969. The thin line is 2013. Okay, so we're going to see like whether there are any changes happening here. Okay, so 4 species. Okay, recorded in 2 surveys. The vertical lines represent the range of heights where the birds were seen on the island. Okay. Yes. So these are the main information. Compare the range in 1969 and 2013 in each of the bird species shown in 5.2. Okay. So we're going to make some comparison here. Okay. So we have some ABCD. We have some discussion for each group of the birds. Okay. This description refer to the changes between 2013 and 1969. Okay, so for A. Okay, for bird A, range extends. Okay, downwards to lower altitudes. Okay, so what can we see here? The changes we can see overall, the activity height is decreasing, has been decreasing. Okay, extend downwards to lower altitude. Okay, so for B, what is going on? The range extend. Okay, extend upwards, higher altitude. Okay, so at this point, we still don't know why. We don't talk about the reason yet. Um we just describe what is going on first. Yeah, so that's always the first step. Describe what is going on, then we start to think about the reason why it could be the case. Okay. Next, C, we can see there are some clear changes there. Okay, the range does not extend downwards as much. Okay, yeah, so we can see this one is quite a big range, but 2013 the range decreased. For D, we can see like almost no changes, little change. Okay, so this is the kind of answer we are expected to give. So to clearly discuss about the changes. Okay. Next. Using the information of figure 5.2, suggest reason for the change in the range of the island's thrust. Okay. So now, okay, we can okay, so these are the changes. Okay, so this is what's going on here. So now we're going to give some potential reason. Okay. Why it could why it could happen. Okay. Okay, decrease in populations. Okay. Competition. Okay. Increase in population, extension of range. Okay. Yeah. So for the ones that decrease, the range decrease, it may be due to decrease in population. Okay. For the one that have increasing range, the range extend, it may be increase of the population. Okay. Another one, competition with other species for breeding nesting site. Okay. So at higher altitude, so this one, the range is decreasing. So this may be due to competition. So other possible reason, habitat loss, deforestation, increased human activity, tourism. So these are human activities that can affect the livelihood of the birds as well. Disease, predation, okay? So here we're trying to explain if there is a change of population, what could cause the change of population? Disease, predation, loss of food sources, okay? Yeah. Loss of food sources may be caused by climate change, global warming, enhanced greenhouse effect, okay, human activities, clearing out the lands, loss of habitat, and so on. Increased hunting could be one of the reasons. Increased pollution, introduction of new species. Okay, so new species of birds present. Okay, so these birds may take up the space, more space. Okay, so the range will be decreasing for some of the birds. Okay, so these are all of the possible reasons that can be accepted. Okay, next. Small oceanic islands are often inhabited by species of birds that are found nowhere else. Many of these species have decreasing populations and are often endangered. Explain the risk to these species of birds that have decreasing populations. Okay, so yeah, so we have like small islands. Okay, so the distance may be very far. Okay, so the birds here, it may be like impossible for them to fly to another island. So these birds here, it may be it just only on this island. It cannot be found anywhere else on Earth. So they are very special and then they can be very important. But because sometimes the environment is changing, so they may be facing decreasing population. Yeah. So what are the risks for them? Okay. Some of the acceptable answers. Decreasing population can have many problems. Okay, difficulty for finding a mate. Okay, inbreeding. Okay, reduce fitness. Okay, yeah. Um, yes, decrease in genetic variation because we are having lower population. So lower population means we have less variety in the genes. Okay, the net genetic variation decrease. Okay, increase in disease, less resistance to disease, more vulnerable to disease. Okay, unable to adapt to change in environment, increased risk of extinction. Natural catastrophes. Okay, so yeah, so if one natural disaster happen, they may be wiped off because their population is so small. Introduce like other species, other competition, okay, they may die off easily or poaching and so on. Okay, so they can face many potential problems when their population decrease. Then they may be at a greater risk of extinction. Okay, next. Okay, so in this question, so this one is about blood circulatory system. Okay, so we have two types of animals here. We have fish and we have mammals. Okay, so if we take a look here, so we can see clear differences between these two types of animals. Their blood circulatory systems, we can see clear differences. Okay, so there are advantages and disadvantages. Yeah, so these are the points we're going to discuss in this question. Okay, next, let's take a look. So first, we need to identify some of the parts. Okay, so first Q, this. Okay, so fish Q. So we can see that, okay, the tubes will be the blood vessels. The tubes will be the blood vessels. So here, this structure with muscles here, straightforward, is going to be the the heart. Okay, the heart that pumps the blood that circulate. Okay, so straightforward. So it will be the heart. T. Okay, here. So here we can see this is the blood vessel leaving the kidney. So first point, we know that the blood vessel leaving the organ, returning back to the heart, we know they are all veins. Okay, returning back to the heart, they are all veins. Okay, they are veins related to the kidney. So there is a specific name for that. We call that renal vein. Okay, related to the kidney. Okay, next, W. Okay, so we can see this blood vessel here. So basically, this is a major blood vessel. Major blood vessel means the veins from all of the body parts, they will um, combine together. When they coming back towards the direction of the heart, they will combine to become a large major blood vessel that return back to the heart. So this blood vessel has a specific name. Okay, so it is the vena cava. Okay. Next, X. Okay. So X. So here we are talking about when we have the deoxygenated blood coming from the different parts of the body returning back to the heart. So the oxygen is already used up by the body cells. So this is deoxygenated blood. Low oxygen. Okay. Low oxygen content blood. So this blood we cannot pump to the body because it's deoxygenated. So we need to return it back to the lung first to replenish the oxygen, to get the oxygen concentration return back. Then we will send back to the heart and then send to the rest of the body. So this is how it happened. So X here is a specific blood vessel that has the responsible to send the blood from the heart to the lung. Okay, so this specific blood vessel is called pulmonary artery. Okay, so it makes sense. Pulmonary means it's related to the lung. Artery means the blood vessel coming out of the heart. They are always called arteries. Okay, the blood vessels returning back to the heart are veins. Okay, so that's the main idea. Okay. State the name of structure V and describe the function. Okay. Okay, so this middle section here that separate the two halves of the heart. Okay. So this is quite important because on this side we have the oxygenated blood. Here we have oxygenated blood. So they should not mix together because we just want the oxygenated blood to go our body. We don't want the deoxygenated blood. So we don't want them to mix together. So this V here is very useful because it separates the two sections of the heart. Okay. Yeah. So what is it? Okay. So basically, V is the septum. Okay. So separates, prevents the mixing of oxygenated and deoxygenated blood. Okay. All right. So figure 6.1. Okay. Just now. Why fish circulation is called the single circulation? It's clear because the main point is the blood passes through the heart once only. Every cycle, it passes through the heart once only. So that's why it is called the single circulation. Blood passes through heart once in a complete circulation, only once. Okay. Next. Advantage of double circulation. Okay, advantage of double circulation. So here we can see for the mammals, for example, the human heart, we have double circulation. So means for each complete cycle, the blood will pass through the heart twice. Okay, so when it's coming back, the deoxygenated blood, it will go to the heart first and then go through the lung and then return back to the heart before it pumps to the whole body. Okay, so this is quite useful. Okay, we will see why. Okay, all of the acceptable answer. So basically, this will be more efficient. Why? Because when you pump to the lung, because we have different parts of the heart, it goes through twice. When we pump to the lungs, we can use low pressure to pump to the lung because the lung is like one organ only. So we don't need the high pressure. So we can save energy. And then the lung is like very thin. The tissue is very thin. So we don't want the high pressure to damage the lung tissue. So go to the lung is low pressure, and then go to the body. Okay, all of the body parts, we will have high pressure. We will have high pressure pumping of the blood. Okay, because we are going to many organs. Okay, yeah, so this is quite useful. Yeah, so because we have the double circulation, we can have high pressure and low pressure situation for different purpose. So it makes it more efficient to achieve different purpose. So we can supply blood, oxygen, and nutrients to the body more efficiently. Okay. At the same time, when we are removing the waste, it is also more efficient. Okay. Yeah. So basically, we will have lower pressure in the pulmonary circuit. So for going to the lung, it will have low pressure. This is quite good because it will be less damage to the lungs, the blood vessels in the lungs. Okay. More time for gas exchange because low pressure, the blood will flow slowly. Okay. So more time for gas exchange, we can get more oxygen. Higher pressure. So for this part here, we have higher pressure. It's more efficient. Okay. For example, filtration in the kidney can maintain high metabolic rate of respiration. So basically, here, high pressure, the body parts can get the oxygen and nutrients faster. So the respiration metabolic rate will be increasing. Yeah. So the main advantages of the double circulation. Okay. Blood vessels that supply the heart muscle can become blocked. Okay. Yeah, so we are quite familiar with this. So blood vessels, okay, especially for the older people, blood vessels can be blocked over time. The name of the blood vessels that supply the heart muscle. Okay, so just now we already talked about the blood vessel that supply the kidney will be um, renal vein or renal artery, and then for the lung is pulmonary. So for heart, they have specific name as well. So for heart is what we call um, coronary. Okay, so coronary means related to the heart. Coronary arteries. Okay, so the heart is pumping blood to all the body parts, including itself. Okay, it's pumping blood to all the different body organs. At the same time, it also have the blood vessel to supply for itself because the heart itself also it need nutrients, need oxygen, yeah, need everything. So it also supply to itself through the coronary arteries. One way which blockages in these blood vessels can be treated. Okay, so yeah, so this is straightforward. Okay, so once the blood vessels already blocked, what can we do? Okay, drug treatment, for example, aspirin, okay, or undergoing surgery, okay, or bypass, okay, to remove the blockages, or we straight away change to new blood vessels. Okay, next, substances move between blood tissues at various sites in the circulatory system of mammals. Oxygen is absorbed into the blood as it passes through the lungs. State the structures in the lungs where oxygen passes into the blood from the air. So basically in our lungs, if we zoom in, we will get to the alveoli. Okay, so these are very, very fine structures where the gas exchange happen. So just now we already mentioned that we send the blood to the lungs to get the fresh oxygen. So and to remove the excess carbon dioxide. So this is how we do it. So yeah. And then we have many of them. Okay, many thousands of them. this is very good because it will provide very high surface area for the gas exchange. Yes, so that's the main idea. Okay, so if we zoom in into one of the alveoli, we can see how the gas exchange can happen. So essentially, yeah, so basically that's our answer here. The structure where how oxygen enters the lung. Okay, so the blood, the oxygenated blood will enter from here. Okay, and then the deoxygenated blood will have oxygen low, and then carbon dioxide will be high. Okay, and then the air in will be the air that we breathe in. Okay, so this air will have oxygen high, carbon dioxide low. So we set up a concentration gradient. So which means carbon dioxide will diffuse into here, and then oxygen will diffuse into the blood. So this is how we can replenish the oxygen supply in our lungs and in the blood, and then we send it to the heart to supply to the rest of the body. Okay. The site of filtration of blood in the kidneys. Okay, where filtration happens in the kidney. Essentially, this is our kidney here. Okay. So the kidney, if we zoom in, okay, if we zoom in into our kidney, we will find this microtubules. Okay, so microtubes. Okay, so the main thing that happens is when blood comes in, it will undergo filtration. So it will undergo filtration. Yeah. So basically, the large particles cannot pass through, like the protein, the blood cells, the big things cannot pass through, but small particles can pass through, like water, sodium chloride, yeah, some ions, urea, okay, and things like that can pass through. Okay, so it will pass through, and then yeah, and then when it reach here, we will start the reabsorption. So because we are losing these things, we want to reabsorb the things that we want. Okay. So for example, we want to reabsorb glucose. Mainly we want to reabsorb glucose. Okay. And then some of the ions. Okay. And salt ions, and then some of the water. Okay. So that's useful. We can reabsorb what we want. What we don't want is it will be made mostly urea. Okay. So urea we don't want. So urea will continue on to become the urine. Okay. Yeah. So the site of filtration will be hold on. The site of filtration will be the glomerulus, okay, or nephrons, which is this part here. So mainly we are talking about this part where the filtration happens. Okay. Next. State the name of the process which products of digestion move into cells and are used to become part of the cells. Okay. Assimilation process. Okay. So what happened is when our digestion already finished, so means all of the large food particles, they are broken down into smaller molecules like the starch, okay, eventually become the glucose, and then protein eventually become amino acids, okay, fats become fatty acids and glycerol. Okay, so these are the large molecules. They are insoluble. Okay, so we cannot use them. But once they are digested into small molecules, they become water soluble, and then they are small enough for us to absorb. Okay, so in our blood, in our small intestine, we have the villi. Okay, so the villi, we can absorb all of this glucose, amino acid, fatty acids. Okay, so once we absorb, okay, we will send to different parts of the body to use them. Okay, so this is what we call assimilation. Okay, so assimilation means it's incorporated into our own body to be used. Okay. Name of the process in which excess amino acids are broken down in liver cells to produce ammonia. Okay. So in our body, if we eat the protein just now, proteins will be broken down into amino acids. Okay. But sometimes when we have too much, we don't store it. We cannot store it. Yeah. So for like fats and sugar, okay, we can store, but amino acids we cannot store. So we need to get rid of it. Broken down in the liver, and then finally it will become the urea to be removed from the as a urine. Okay, excuse me. So we will this process will happen in the liver, breaking down process. So this is what we call deamination. Okay, what we mean by deamination. Okay, we can have a quick look. Yeah. So this is the structure of the amino acid. Okay. So structure of the amino acid. So we can see it has three main parts. So any amino acid will have amino group, which is NH2, and then it also has carboxyl group, which is COOH. Okay, all of the amino acids will have these two structure, and then they always have hydrogen atom here, always have one. And then on the other side, they will have the R side chain. So the side chain, it can be anything. It can be like another carbon chain, or it can be like some like ring structure, something like that. So it can be like other things. Okay. So that's why we have like 20 different amino acids in our body. So 20 amino acids, their difference is in this side chain here. The rest are identical. So the difference in the side chain produce the 20 different amino acids. Okay. So in our body, when we have too much amino acids, we want to get rid of it. Okay. But, okay, let's take a look. We want to get rid of it, but we don't want to like throw away the whole thing. We only want to get rid of the things that we don't want. Okay. So for example, this part, hold on. Okay. For example, this part. Okay. So this part, maybe we have some like carbon, oxygen, hydrogen. Actually, this part we can use for the respiration. We can produce energy. Okay. So this part we will keep. This part, the amino group, is not that useful to us. Okay. So basically, the idea of deamination is remove the amine group. So in the liver, it will remove this amine group. Okay, it will keep this part. Later, we can use for energy respiration. Okay, but the amino group, we will cut off. Okay, and then after that, it will turn to urea. Okay, yeah. So this is how we have the, this is why we have the deamination. Okay, so actually our body is a very, very efficient system. It will not waste anything if possible. So for this excess amino acid, it will just cut off the amine group and then remove as the urea, but will keep this part, okay, for respiration purpose. So that's the idea. Okay, next, state the name of the organ that releases estrogen into the blood. Okay, so we know estrogen is a hormone, okay, related to the female reproductive system. Okay, So related to the female reproductive system. So what will be the organ that releases it? Okay, so it is the ovary. So the ovary is the main organ that that produce and release the estrogen. Yeah. Okay. So with this, we finish the paper.